Hello everyone. Today we will study what is linkage. In the last lecture we studied the dihybrid cross, and based on the dihybrid cross we studied the law of independent assortment. According to the law of independent assortment, we have seen that two different genes, which are controlling two different characters, always assort independently, right? But is this applicable to all the genes? Okay, now in case of pea plants, we studied the cross considering seed shape and seed color. And we saw that the gene for seed color and shape assort independently. Now the question is it applicable to all the genes in all the organisms, right? And when similar hybridization experiments, just like the pea plant done by Mendel, similar experiments were done in different organisms, it was found that when certain dihybrid crosses are made, considering some characters, a pair of characters, that time independent assortment is not seen. The typical ratio 9 is to 3 is to 3 is to 1, which we got in the Mendelian dihybrid cross, is not seen in every cross. That means there is some exception. And that exception to the law of independent assortment is linkage. So in this video lecture, we will study the concept of linkage and a very important topic, that is Morgan's experiment, which actually led to establishment of this particular concept of linkage. Okay. So first, what is linkage? Now, see, if genes are present on two different chromosomes, okay, then obviously as different chromosomes assort independently, the genes present on different chromosomes will also assort independently. But if the genes are present on same chromosome, that time all the genes which are present on that same chromosome, right, a particular chromosome, will get inherited together. Understand? One chromosome, like one chromatid, is one DNA molecule, one double-stranded DNA molecule, and all the genes are present on it, right? In this case, what do we see? That if the genes are present on a single DNA thread molecule, and this DNA will go in either of the gametes, right? So, the genes present on the same chromosome get inherited together. So, how can we define linkage? Let's write the definition first. The tendency, the tendency of gene present on same chromosome, same chromosome to get inherited together, right? To get inherited together. This is linkage. This is linkage. Okay. Let's take an example to first distinguish between independent assortment and linkage, and then we will also study the Morgan experiment. Okay. So let's say first, let's take a pair of genes. Okay, A and B. First scenario, let's say this is a homologous pair of chromosomes, and let's say it has gene A here, and let's take the contrasting allele small a on its homologous pair. We have another pair of chromosome here, let's say like this, okay, and it has gene B. Let's write small b, okay. So we have gene A on the first homologous pair and gene B on the second homologous pair. Here we have considered genes are present on different chromosomes. So what will happen here? As per the law of segregation, this homologous pair, so these alleles will also separate during gamete formation. Also, this capital B, small b, this will also separate. Now, this first chromosome, right, first yellow can go with first blue, or this first yellow can go with second blue. This is only the independent assortment. There is equal chance, right? Also, this first yellow, the second yellow can go with the first blue, or the second yellow chromosome can go with the second blue. So accordingly, we get different combinations. So if we represent all the genes. What do we get? First gamete will receive capital A, capital B. Next will receive capital A, small b. Next one will receive small a, capital B. And the last one we have small a, small b. So what do we see? There are four different types of gametes and all these gametes will be produced in equal proportion. So as there are four types and equal proportion, so 25% each. Okay, this is the gamete distribution. Okay. Now here as the gene pair A and B are getting assorted independently. So simply this is explaining the law of independent assortment. Independent assortment. Okay, this is like what we studied in the pea plants also, in the Mendelian dihybrid cross. Now let's see what happens actually in case of linkage. Let's say same gene A and B but now present on same chromosome. So let's say this is the chromosome. Okay, homologous pair of chromosomes. Here we have gene A. So on the same locus we will have its allelic form. Okay. Let's say here on the same chromosome we have B and here we have small b. Okay. Here gene A and B are present on same chromosome. Again, law of segregation, what will happen? This homologous pair will separate. So capital A and small a will separate. Capital B and small b will also separate from each other. Now, what about the segregation part of chromosomes, the gene pair? This chromosome will be in the 50% gamete and this chromosome will be in the remaining 50% gamete, right? So you have capital A, capital B type of gamete and small a, okay, we have small a, small b type of gamete, okay? Now, this is basically like these are the two gametes which are formed directly by segregation of this homologous pair. So these two will be called as parental type of gamete. Okay, there is no change, right? As it is, the chromosomes have separated. So parental type of gamete. Okay, now there is one particular scenario we study in cell division topic, right? In meiosis, basically in Pachutine phase, crossing over occurs, right? There is exchange of genetic material. So it may happen that actual crossing over occurs between the gene A and B. So that time in that particular scenario crossing over, this capital A, like this part, let's say, will move here and this small a part, this will move here. So there will be exchange of genetic material. Okay, and let's say the exchange is right between genes A and B. So this capital A moved here, small a moved here, and then the chromosome separated. In that case, we will get, like, imagine this small a and capital A. Okay, these are interchanged like this. What we will get? We'll get small a, capital B type of gamete and we will get capital A, small b type of gamete, right? Now, as these two are formed after crossing over, okay, after recombination, we call them recombinant gamete, recombinant type of gamete, okay? Now, these are the four types of gametes, same as we have seen in the first case here of independent assortment, but as the genes A and B are present on the same chromosome and we have also drawn it very close, right? Both genes A and B are very close to each other. So, what is the chance that crossing over process occurs right between these two genes? Obviously, it will be rare, right? Most of the cases, what will happen? The chromosomes will separate as it is and hence capital A, capital B will remain together and small a, small b will remain together. But sometimes, rarely, when crossing over occurs between these AB genes, right? That time you will get these two types of gametes, the recombinant type. So always, if the genes are present on same chromosome, you will notice that the parental types of gametes will be in more proportion. Okay, and the recombinant type of gametes will always be in less proportion. Okay, Now, as we see here, we can get four types of gametes, but the parental ones, okay, same, like the genes were present in parents, same type of genes, the same gene combination is present in the gamete and hence the offspring. So these will be the parental type, this will be more. And if suppose crossing over occurs, recombination occurs and then the gametes are formed, you get the recombinant gametes. Now crossing over will be rare, especially if the genes are very close to each other, right? What is the probability that crossing over occurs exactly between the two genes which are very close to each other, right? So it is very less. So that is why less number of recombinant gametes. In this case, what we have noticed that the genes present on same chromosome, like if you take the first chromosome, the gene capital A and capital B were present on same chromosome. So there was a tendency that this capital A and capital B will stay together, right? Also, small a, small b are together. So there is a tendency that is small a, small b will remain together. There are more number of gametes of these types. And sometimes rarely when crossing over occurs, you get this. And that is why we say that there is linkage. The capital A and capital B, they try to stay together because they are on the same DNA thread. They are on the same chromosome. Here comes the concept of linkage. And we define linkage using this only, right? What did we say? The tendency of genes present on same chromosome to get inherited together. So this is linkage. Now let's understand the Morgan's experiment, okay, which actually established the concept of linkage. Okay, the term linkage itself was given by Morgan. Okay, the scientist name first very important. We have Thomas Hunt Morgan. Morgan. Okay, for his experiment, he took an organism, a very simple organism, fruit fly, that is Drosophila, Drosophila melanogaster, melanogaster. This is fruit fly. Okay, now the question is, we have so many different types of organisms. Why fruit fly? Right? Basically, it's a tiny organism, easy to grow in small space. Okay, like in Mendelian experiments, pea plants were grown, so you require a huge area to grow hundreds and thousands of plants. But in case of Drosophila, these are tiny flies, so obviously you can easily grow them in small containers, like small flowers in a small laboratory. Okay, next, it has few number of chromosomes, only four pairs of chromosomes. So it is very easy to assign genes to specific chromosome. Like this gene is present on this chromosome. It is easy to assign because there are few number of chromosomes. Also, we have separate male and separate female Drosophila. So here basically sex-linked characters were studied by Morgan. How sex-linked characters, basically the genes present on X chromosome were studied. That is why we say sex linkage was studied by Morgan. Okay, and when we see the cross, we will see a particular pattern, like a crisscross pattern, which is seen in case of X linkage. Okay, so that is what is studied in case of Drosophila. Okay, so these are tiny flies, easy to grow, it has less number of chromosomes. We have easily distinguishable male and female Drosophilas. Okay, plus the medium required to grow is also very simple, right? So because of all these characteristics, and one very important thing, right, which will help us to do experiments, that is Drosophila has easily observable contrasting characters, like we had in the pea plant, right? Pea plants were either tall or short, with round seeds or wrinkled seeds. So easily observable contrasting traits. In the same way, in case of Drosophila also, we have many contrasting traits, okay, easily observable. And that is why Drosophila was chosen. So Thomas Hunt Morgan studied on Drosophila melanogaster, that is fruit fly, and carried out some dihybrid crosses. Now, which characters were considered? So, let's write this first, okay? Before studying and making the actual cross, make sure you know all the characters which were studied in Drosophila and also which character is wild type dominant and which are the mutant type recessive. Okay, so here we will directly draw the table so that you can easily remember all the characters. So first, let's see what different characters were studied. We have characters. Next, we have the dominant type. Now, in this case, the dominant, okay, these were like the original phenotype. So, these are called wild type. What are these? Wild type. Okay. It is not necessary that wild type should be dominant only, but in most cases it is dominant. So in case of Morgan's experiment, whatever three characters we will see, in all the three cases, the wild type is dominant. And then what will be the contrasting? We will have the recessive, that is mutant. Okay, we have recessive. Okay, let's write all the characters. First, we have body color. What do we have? Body color. Okay. Now, in case of Drosophila, the body color are of like two different types, contrasting, brown and yellow. Okay. The wild type dominant is brown, and the recessive form, the mutant form, is yellow. Okay. We have yellow. Now, usually in Mendelian cross also, what did we see? That whatever is the dominant character, its first letter is taken. So, as per the typical Mendelian type of cross, we would have written brown capital B and yellow small b, right? But there are some other ways also to represent genes. So the way which we use here is we take the mutant type, the recessive type. Okay, take the first letter of it. Okay, so yellow is the recessive. So yellow will be represented by Y, and the dominant one will be mentioned as Y plus. Okay, plus represents the wild type. Plus is wild type. So Y plus. Okay, so this is as good as capital Y, Y plus. Okay, and small y is as usual recessive. But make sure here we have taken the first letter of recessive character. Next, we have eye color. Okay, eye color. Your dominant is red and recessive is white. Okay. So which letter should we use? Obviously recessive first letter, white W, and in case of red we have W plus. Okay. Next, third character, we have the wing size. Wing size. Either we could have normal size or the miniature, mini size, small size. Okay. Miniature. So again, first letter of the recessive trait. So M will be miniature and normal will be M plus. Okay. So these are the various characters which were considered by Morgan in Drosophila, and all these characters, okay, body color, eye color, wing size, okay, each one is controlled by a separate gene, and all these genes, okay, genes for these three characters, okay, are present on X chromosome. Genes are present on X chromosome. Very important. That is why we say it is sex linkage. Okay. All the genes which were studied, their inheritance, these are present on X chromosome. Okay. So these are the characters. Okay. Remember this first before trying to study the cross. So body color, we have brown or yellow. Also remember the letters. Next, eye color, red or white, wing size normal or miniature. Okay, now let's see the crosses which were done by Morgan. Okay, so in your textbook, basically these crosses are shown like this. So we have two crosses over here, as you can see, right? We have cross A, here we have cross A, cross A, and we have cross B. Okay, these are separate crosses. First, let's focus on the first cross. Okay. So as you can see, these we have female and then male Drosophila, right? In case of the fertile, male and female Drosophila. Okay, these are the chromosomes. Okay, like in female, you have like X chromosome and X chromosome. Okay, and others will be autosomes. And in case of males, we have X and Y like this. Okay, these two are X chromosomes and these is X and Y chromosome. Though the method of sex determination in Drosophila is not same as humans. In humans, we have XX, XY type of sex determination. In Drosophila, we consider ratios, okay, between the sets of autosomes and the number of X chromosomes. So that is a totally different sex determination method. But the fertile Drosophila which were used here, right? The Drosophila to reproduce should be fertile. We can't take sterile ones. So fertile ones, here we see that apart from autosome, we have these two chromosomes. In female, it's XX and in males, it's XY. And the genes which we are studying, these are present on which chromosome? We mentioned it earlier, X chromosome, right? So look at the letters. We have Y, W, right? Y plus W plus. That means which two characters are considered here? Dihybrid crosses are done. So at a time we will consider only two characteristics, right? So as we have used Y is yellow and opposite character is brown, right? Contrasting. So this is the body color. For cross A, we have considered body color and as we have W, so white and red contrasting. These were eye colors. Eye color. Okay. Now look at the second cross. Here, what letters we have mentioned? W and M, right? W is the same W here, okay? So W is eye color, and what is M? M is the miniature size of wing, and the M plus will be normal. So this is wing size. So in the cross B, this is also a dihybrid cross, but here the character which we considered, okay? This is wing, this is wing size. Wing size. Eye color and wing size. Okay, so these are the two dihybrid crosses. For cross A, we consider body color and eye color, and for cross B, we considered eye color and wing size. Now, what I will do is, I will make this cross A in detail, and then we can easily compare the cross B with it. Okay, so let's draw one by one. We have cross A considering body color and eye color. So, first let's take the female Drosophila. We have body color and eye color. Now see here. Here we have Y, Y, W, W. Okay, and we have Y means recessive, so yellow body color, and W means again recessive, so white eye color. So I will draw it again. Let's say this is the X chromosome. We have Y, W. Another X chromosome in female. We have Y, W, right? And we also have the centromere. This is like XX chromosome in female Drosophila. Next, it is crossed with the male. So in male, now here I will use a different color so that we can distinguish between the chromosomes also. So let's say this is the male Drosophila's X chromosome. And so again, it will have the same genes at same location. But here Y plus W plus, right? What do we have? Y plus W plus. As we have plus both the genes. And the other chromosome is Y chromosome, okay? As Y is a totally different chromosome, it will have different set of genes. It will not have Y, W or any other, the same genes as X chromosome. This has totally different genes, okay? This is the Y chromosome, okay? Not Y gene, Y chromosome. So it has totally different set of genes. So male has X, Y. Now here, get the gene. Look at the gene, Y plus W plus. As there is only one X chromosome in the male Drosophila, whatever genes are present on this chromosome, dominant or recessive, it will be expressed. Okay. If there is only one copy, then it is called hemizygous. Usually we use the term homozygous and heterozygous, right? But to say any genotype, homozygous or heterozygous, first of all, we should have the alleles in pairs, right? Like if we have the identical copies, we call it homozygous. If dissimilar copies like capital T, small t, then heterozygous. But what if there is only single copy? Like the male Drosophila, as there is only one X chromosome, it will have only one copy of all the genes on X chromosome. Like here, one copy of Y plus, one copy of W plus, whereas female has Y, Y, W, W, right? So this condition wherein a single copy is present of the genes, it is called hemizygous. Okay. Now these are the parents. What are these? These are the parents. So what is the first step now? You have to write the gametes, right? So let's write the gametes. So in your textbook, directly the F1 generation is shown. We will write each and every step to understand how actually we got the F1 and F2. Because sometimes it becomes confusing. Like how do we get like all these F1 and F2 offsprings, right? How they have mentioned the results directly. Basically, if we draw each and every gamete, it's easier to imagine. So now here, these are the XX chromosomes. What will happen during gamete formation? Meiosis. So the homologous pair of chromosomes will separate, right? So these two chromosomes will separate. So whether you take the first one or the second one, does it create any difference? No, because on both the chromosomes we have same set of genes, Y, W, Y, W, right? So here only one type of gamete is produced. That means all the female gametes produced will have this YW. Okay, one type. Also, let's consider rarely whenever crossing over will take place. What happens during crossing over? This Y, okay, let's say this much part, okay, let's say a different color. Let's say this part is exchange. Will it create any difference if this Y comes here and this Y goes above? Does it create any difference? No. When the chromosomes will separate, you will have IW. So here the female is producing only one type of gamete. What about male? Now see for males, as we have one X chromosome and one Y chromosome, and here crossing over will not take place, okay? So they will separate as it is always. So for male Drosophila, you will always have two different types of gametes, whatever may be the genotype, dominant, recessive, hybrid, whatever, okay? You will always have the two types of gametes, okay? So let's write the two types of gametes. What are these two types? One with the X chromosome, so we have Y plus, W plus, and the other with, other 50% with Y chromosome, okay? See, X chromosome and Y chromosome, right? So one type of female gamete and we have two types of male gametes. The female gametes are with the X chromosome and obviously other autosomes are also present, but we are not studying the genes present on other chromosomes. That is why we have drawn only the chromosomes concerned in this experiment. Other chromosomes will also be present in the gametes. Okay. So we have one type of female gametes, two types of male gametes. Now gametes are done. What is the next step? Fertilization. So either these two will fuse or these two will fuse. So accordingly we will get the gamete. So let's write the offspring. First we have here Y, W and this male gamete. So we have Y plus W plus, right? And what about the second one? We have the X chromosome from female, Y, W, and the other one is Y chromosome, right? Here we have XX, so obviously this is female offspring, and here on the right hand side we have the male offspring, male Drosophila. Okay, this is our F1, first filial generation. So we have the parents, we have the gametes and F1 generation. Okay, 50% females with this genotype and the remaining 50% males with this genotype. Okay, now let's assign the characters also. We have not mentioned the characters. Now here in the first case, we have YY. Okay, YY, this is recessive. So what is the recessive body color? What is the recessive body color? Yellow. So this female is yellow body color and WW means white, right? The earlier table which was white eye color. What about male? As there is only one copy, it will be expressed. Hemizygous, so whatever copy is there, it will be expressed And here we have Y plus W plus, the dominant copy, okay, wild type. So this is wild type, okay. That means brown body and red eye. Now, what about the offspring? See here, this offspring has genotype like this. Y plus, you can write it like this. Y plus Y, W plus W, just like capital Y, small y, capital W, small w, right? In similar way. So, what will be the character expressed? As there is dominant copy plus plus for both, the character will be wild type. Character will be wild type, right? What about male? It has one copy, hemizygous, right? One copy, Y, W. So whatever character is there, it will be expressed. Whatever genes are there, it will be expressed. So now your male Drosophila has yellow body and white eye. Okay, what do we see? I see in the parental generation, whatever was the character of female, right? Female parent. Now we see it in the male offspring. Okay, and whatever was the character of the male parent, it is present in female F1 generation offspring. See, female parent to male and male parent to female. So there is some crisscross inheritance. That is why Morgan said it is sex-linked character. Okay, just directly, how can we know that the genes are present on X chromosome? They are sex-linked. We see this pattern, crisscross pattern. Okay, so now we have the F1. Now, as Drosophila are separate male, separate female, unisexual. That is why we cannot do selfing. In pea plants, the flowers were bisexual, so we carried out selfing. Here we can't do selfing, but we have the female F1 and male F1, so we will do intercrossing between these. Okay, so we'll take this female and cross it with these males, and then see what are the offspring. Okay, so first thing, what will be the gamete? Okay, so let's try to figure out the gametes first. I will draw the F1 generation female here. So we have Y, Y, W, and the other chromosome, it will draw in blue color. Why blue? Because it came from the original male parent, right? So we have Y plus W plus. And obviously all the chromosomes will also have the centromere. Okay. Now we have here F1 female. Okay. Let's understand how many different types of gametes will form. So what will happen during meiosis? Again, meiosis will take place to form gametes. So this chromosome, the yellow chromosome and the blue chromosome will separate, right? So let's say if they separate as it is without crossing over, what we'll get? We'll get first gamete which has Y, W, okay? And then we will get another wherein we will have this blue chromosome, okay? Y plus W plus, right? Let's say the chromosome separated as it is. Okay, so 50% has this, 50% has this. But there is also a chance that crossing over takes place between these Y and WG. Okay, there is exchange of genetic material between these two chromosomes. So that time, this Y will move here and this Y plus will move here, right? After exchange. So that time you will have new combinations of gametes. So let's draw it. So it will be like this, like half yellow, half blue, right? Let's draw it here. So let's say we have this Y plus moved here. So we have Y plus and then this original W. Okay. Then the other remaining we will have some parts of yellow chromosome and the remaining blue. Let's write the genes Y and W plus. Okay. So first two gametes which are shown here. These two. These are produced without crossing. Directly the chromosomes have separated. So these are like parental. As they were present in parent, full yellow or full blue, so they are present like this, right? Whereas in this case, these two, what we have seen, these are produced after crossing over, after recombination. So like you can see, like some blue part, some yellow part, some yellow, some blue, right? So these are the recombination. So obvious these two top two will be in more numbers, and when rarely crossing over will take place, we will have these two which will be in less numbers, right? We have four types of gametes, but out of these top two are more in number, and these bottom two will be less in number. What about this male? This male is crossed, right? We saw this female and what gametes are formed. What about this? Okay, simple. As this is male individual, the X and Y will separate as it is. So simple, 50% of the male gametes will have this yellow chromosome, X chromosome, and the remaining 50% will have Y chromosome. So here we have four different types of female gametes and we have two types of male gametes. Okay, so 4 into 2, we will get 8 offspring. And now we have to see what are these 8 offspring. Okay, so let's draw another table to understand this. I will write all the gametes. See, if you draw this by your own hands and to understand, then once you're sorted, once you can visualize everything, is very easy. So we will draw everything step by step. Okay, let's mention all the gametes. This is the next step. So here we will mention the male gametes for F1 and here we will mention the female gametes of F1. Okay. Female gametes are of 4 types and male gametes are of 2 types. So let's write it. Okay. So what are the female gametes? See here. These 4, right? These are the female gametes. Full yellow, remember with color. Full yellow, full blue. We have some blue, then yellow, and then yellow blue. Okay. And male, we have these two types of gametes. Full yellow and then Y chromosome. So let's write it. Female gamete, full yellow, Y, W. Then we have full blue, Y plus, W plus. Next, we have some blue part and then the yellow part. Right? Let's write the gene Y plus and then remaining part will have W, right? Next, we have some yellow part because of crossing over and then the blue part. In yellow part, we have the Y gene and in the blue part, we have W plus. These are the 4 female gametes. Now, what about male gametes? We have full yellow, Y, W, right? And then we have the Y chromosome. Okay? These are the gametes. Let's quickly find out all the offspring. Okay, this is our Punnett square. 4 female gametes, 2 male gametes. So 4 into 2, we will get 8 offspring. We will write all the offspring here. Okay, simple. You have to do match the pair as we did in the monohybrid and dihybrid process. If this female gamete is with this male gamete, you have to write it here. If this female gamete is with this male gamete, you have to write it here and so on. So I will write it quickly. Okay, so we have Y, W, and the other one is also with Y, W. Right? Easy. YW, YW. Next, now we have this female chromosome. So let's write it. We have Y plus W plus. Okay. And next we have this male chromosome. Y, W. Okay, so accordingly, let's copy the entire thing. Half blue, half yellow, so recombinant. We have Y plus and then W, and now copy the male chromosome. Now last, half yellow, remaining blue, right? We have Y. Then in blue part we have the plus, wild type. So W plus, then we have the male gamete, copy from top, YW. Okay, these are the offsprings. Now copy here. Okay, here. So these female gametes is fused with the male gametes carrying Y chromosome. We'll write it here. So I will quickly fill this. Half blue, right? Half blue, and then yellow. Write it Y plus W. And then the Y chromosome. And last, yellow, blue, we have Y, and then W plus another Y chromosome. Okay, so we have all the offspring. Now what do we see? Now very important. This analysis is important. The male gametes will be in equal proportion only. Okay, so this is 50%. This is also 50% gamete. What about the female gametes? As these two, full yellow and full blue, these are parental. So these will be in more number. And these two, the recombinants like half yellow, half blue, these are recombinants. So these will be less in number. Okay, we have just drawn these four types of female gametes and two types of male gametes. But remember, these two types of female gametes will be more in number and these two will be less in number. Both the male gametes will be equal. Okay. So if you look at these offspring here, these, here, okay, these offspring, four. What can we see? As these two female gametes will be more, these four types of offspring will also be of like more frequency. They will be and more frequency, right? More number. And what about this one? Let's do this. These four as female gametes are equal. Okay, so now these two female gametes, as these are less in number, these are recombinant, so less in number. So these four types of offsprings are also less in number. Correct? And when Morgan carried out this entire experiment, he found that when he took these two characters, body color and eye color, that time the recombinant ones, okay, these, okay, now how do we know that these are only the recombinants? We can't see the genes directly. So we can see the characteristics, okay, as these are like half blue, half yellow. So we see some new combinations of characters expressed in these four offspring, the bottom four. Let's write the phenotypes of all. Quickly, let's write the phenotype. So, as this is YYWW, all recessive, so this will be yellow, white. What about this male? Y chromosome does not have the body color and eye color gene. So, just X chromosome will decide this one chromosome. So, again, yellow, white. Okay. What about this? Y plus Y, W plus W. As there are plus plus, both Y plus as well as W plus means dominant characters will be expressed. So this is wild type. Okay. Wild type. Same here, Y plus W plus, so wild type. Okay. What about this one? Now here, very important. Y plus is present. So the body color will be wild type. It is brown. Okay. And the eye color, WW. So eye color will be recessive one, that is white. Okay, eye color is white. Okay, what about this one? We have white plus the same thing here. Brown body and white eye. What about this one? Exact opposite. We have YY, so yellow body. We have yellow body, recessive. And then W plus is present. So eye color is wild type, red, as we saw in the table. Same here for male, yellow, and we have W plus, so red eye color, right? So we have mentioned all the phenotypes also. So let's go back to the parental generation. Here, here. This is where we started the cross. These are the parents, right? So how are the parents? Yellow, white, and then wild type. Correct? Original parents now. Here, these four. One, two, three, four. Yellow, white, wild type, wild type. Means just like the original parent from where we started the cross. This is what Morgan observed. How many flies, how many Drosophila in the F2 generation are same as the parent? And he counted. Okay, let's say he has 1000 Drosophila. He counted. Literally you have to count one by one. How many Drosophila have yellow body, white eye? Male, female, okay, both. And how many are like both wild type characters, brown body and red eyes. These were counted. And then recombinant. Now see, there was no fly in earlier generations with brown body and white eyes or with yellow body and red eyes. These are some new combinations which are because of crossing over which occurred during gamete formation in F1 generation, right? So these are new combinations. So that is why these are recombinant. These are recombinant, new combination. Whereas the one which matched exactly like parents, these are parents. Okay, now see everything is tallying here. These four types of offspring, these two females and these two males, these are same as parents and these are more in number. Also, we have seen that these two types of gametes are also produced more in number, right? So now we have to only see what's that number. Okay, more number means what? When Morgan carried out the experiment, he saw that the parental ones here were 98.7% and the recombinant ones were only 1.3. So addition is 100%, right? All flies together 100%. Recombinants are just 1.3% and the parental ones are 98.7%. Okay. So now don't break your head over like from where this 98.7 came. This is an experimental value. If you take Drosophila and start with the cross like this, okay, you take the parent with these characters, female, yellow, white, male, wild type, obtain the F1, then obtain F2, okay, and then count that in F2 generation, okay, how many have parental and how many have recombinant characters. Okay, if you count it, you will see that in F2 generation, second filial generation, 98.7% have the same body color and eye color as the original pair from where you started the cross, and only 1.3% were recombinant type. Okay, these two females and these two males, recombinant 1.3%. This is an experimental value. Okay, now as the recombinants are so less, it suggests that the chance of crossing over between the gene Y and W is very less. That is why you are getting only 1.3% recombinant. The chance of crossing over between body color and eye color genes is very, very less. Okay. Now let's go back to the NCRT table. Here they have mentioned directly the parental generation, right? Next we have the F1 generation. We did intercrossing between this, and then we got the F2. Okay, look at the F2 generation. Can you see F2 generation here? This. This. Okay. Now see. we have the F2 generation. Okay. Yes. F2. Now the way I drew the F2 generation unit square, what I got was something like this. Okay, these top four were parental and these bottom four were recombinant because the way I wrote the gamete. In your textbook, directly all the parental and recombinants are given as list. Okay, so here parental are like these. So just look at the characters. Wild type, right? Wild type, yellow, white, wild type, yellow. Correct? And also see the chromosome. Full blue, full yellow, full blue, full yellow, yellow, yellow. So no recombination, right? So these are the parental. What is the percentage? 98.7. Whereas look at the recombinant. Okay, we have new combinations. Now they have mentioned only the recessive character. If you have Y plus, obviously it means the Y type body color, right? So accordingly, you can see these are recombinants. That is why you see in all these four offsprings here, you have like half blue, half yellow, yellow, blue, blue, yellow, yellow, blue, right? So new recombinants after crossing over. And these were found to be only 1.3%. Okay, so this is an experimental value. Now, the way we carried out the entire dihybrid cross, considering body color and eye color in Drosophila, in the same way you can take other characters like here. Let's erase this. Maybe. And then we will analyze the characters. Okay. So now look at cross B. Cross B. Let's compare both the cross. Cross B, here we have W and M. So W is for eye color, right? And M is for the miniature wing size. M plus will be normal wing size. So here cross B is also dihybrid cross considering eye color and wing size. First thing which you can note directly, compare both. The distance between Y and W and here the distance between W and M, right, is different. The distance between W and M is more as compared to Y and W. If the distance between W and M is more, obviously the chance of crossing over between these two will be more. Imagine, like, let's say these are chromosomes. You have one gene here, other here. This is homologous pair. What is the chance that crossing over occurs exactly between these two genes? It will be rare. But if you have, let's say, same two chromosomes, one gene is here, other is here. Okay, this is a homologous pair. Genes are away from each other. So somewhere in between the crossing over will definitely take place, right? So that is why you see now in the same way you have like the parentals, the F1 generation. Now look at the F2 generation. This is important. Look at the F2 generation. See this F2 generation. You have to do the cross in same way. But look at the F2 generation, parental and recombinant. Okay. As you can see here, parental type for only 62.8% and recombinant 37.2%, right? So recombinants are less in number as compared to parental. That means there is some linkage. But if you compare it with first cross, what do we see? The number of recombinants is more in cross B as compared to cross A. Okay. So as recombinants are more relatively more in the second cross, the distance between W and M will be definitely more, which you can see in the cross. Okay, here also you have like these parental and these recombinants. 62.8% parental and 37.2% recombinant. So based on these two crosses, if you compare, what can you say? As you got more recombinant in the cross B, so the distance between these two genes, that is eye color and the wing size, will be more as compared to the distance between the body color and eye color. And this is also mentioned here. Okay, this is a very important line. Mark it in your textbook. Here, can you see this? Here, mark it. The strength of linkage between the Y and W is higher than the W and M. So overall, it's like this. You have one chromosome. There is only one chromosome here. Okay, I'm drawing. X chromosome of Drosophila. It has Y gene over here for body color, W gene over here which is very close, and M gene over here. And you have the centromere. Okay, so distance between these two is less and these two is more. So number of recombinants is also more in the second cross as compared to first cross. But it is not more than 50. Okay, see, recombinant percentage will never be more than 50. The maximum possible percentage is itself 50. And 50% recombinants means independent assortment. Equal chance, right? So simple, we will also mention this very important, but first mark this point, the strength of linkage between Y and W is higher than W and M. Okay, so very important point. We are done with the cross. Always the recombination frequency which we get. Okay, recombination frequency. Okay, very important point. Recombination frequency. In case of independent assortment, independent assortment, the recombination frequency will be 50%. Okay, 50%. In case of linkage, it will be less than 50%. Okay, recombination frequency crossing over chance, like separation of the gene. Independent assortment, it will be 50 to 50% recombinant, 50% parental type. That is why we say independent assortment, right? Whereas in case of linkage concept, we will have the recombination frequency as less than 50%. Okay, so we can also say recombination frequency is less than or equal to 50%. Always, it will not be more than this. Okay. Now, this is from your NCERT textbook itself. The concept of linkage and recombination. All the important points are highlighted so that you can focus on these points. As you can see here, how it's mentioned, the genes which were studied, right? These are present on X chromosome, right? These are present on X chromosome, body color, eye color, and wing size. Also, now see, if there is physical association, okay, or linkage of two or more genes, okay, whatever, two or more genes, okay, we call it linkage. And the term linkage itself was coined by Morgan, C.H. Morgan. And you can see this is a physical association of genes on a chromosome, as the genes are present on same chromosome. An exact opposite concept will be the generation of non-parental gene combination, right? This will be recombination. Simple. Generation of parental type linkage. Generation of non-parental means recombinant types are generated. That will be due to recombination. Also, now based on this recombination frequency, okay, what percent is the chance of crossing over? You can actually draw a map of different genes present on a chromosome. Now this does not show the actual distance, but these maps, the genetic maps are drawn based on recombination frequencies. So we have very important scientist here. You can see it's mentioned over here. Alfred Schwartz, right? He basically first used the frequency of recombination between gene pairs on the same chromosome as a measure of distance between genes and mapped their position on the chromosome. Okay, and that is why we could draw it like this. This is a chromosome. Then okay, gene Y is here, gene W is very close, gene M is a little far from, right? We could map the genes on the chromosome, okay, because of the recombination frequencies. Okay, and you have to know the recombination frequencies. Okay, the experimental data. So here we have completed the linkage topic. I hope this entire video lecture was helpful for you. Thank you.